All right, good morning, everyone. Welcome to the Illinois Soybean Association's Soy Advisor webinar. I am Dr. Stacy Zuber. I'm the research data scientist at the Illinois Soybean Association. Today we have Dr. Matthew Hudson and Dr. Andrew Skaboo, who are going to be presenting combating soybean cyst nematode with advancements in genomic and breeding technologies. This webinar is being recorded. The recording, as well as more information on ISA funded research projects, can be found at ilsoyadvisor.com. That is ilsoyadvisor.com. Throughout the webinar, you can submit questions in the webinar Q&A at any time. We will be, because this is having two presenters, we will be doing questions for the first presentation in the middle, and then we can also address more questions at the end if those come in throughout the second presentation. So I'm pleased to introduce today's speakers. We have Dr. Matthew Hudson. He's a professor of crop science at the University of Illinois with affiliations also to the Center for Digital Agriculture, the National Center for Supercomputing Applications, Institute for Genomic Biology, Center for Advanced Bioenergy and Bioproducts Innovation, and Departments of Entomology and Bioengineering. His research focuses on the application of digital and genomic technologies to problems in crop science, including the discovery of mechanisms and new strategies for SCN resistance in soybean and SCN virulence. Dr. Andrew Skaboo is an assistant professor at the University of Missouri in the Division of Plant Science and Technology, and he leads the Northern Missouri Soybean Breeding and Genetic Research Program. Dr. Skaboo received his undergraduate and graduate degrees at the University of Arkansas and Tennessee, and was a research geneticist with the USDA ARS at NC State University before joining the University of Missouri. His program focuses on both conventional and herbicide-resistant cultivar development, germplasm enhancement, and research in the genetic architecture and underlying important agronomic and seed quality traits, including soybean cyst nematode, as well as seed protein and oil content. We're going to start the presentation today with Dr. Hudson, so I'll go ahead and turn it over to you to introduce today's topic and begin your presentation.
Hi, everyone. I want to apologize in advance. We're in the middle of a hailstorm here, so if you hear strange noises, that's hail hitting the window. And I hope my power and my internet stays on for this, but at least there's no soybeans out there yet. So, so I'm going to talk a little bit about my work, which, as Stacy kind of introduced, is kind of a mouthful. But the genomics and molecular biology of soybean and soybean cyst nematode and how those two interact a little bit. And a little bit about the digital agriculture side of things and how we think that AI and digital technology is going to help with a few of these things. And then Andrew's going to give you more of an in-depth picture of where we are right now in terms of SCN resistant soybeans and the things to do and not do. So, as I mentioned, I'm really going to talk here about genomes and molecular biology. This work is funded by Soybean Checkoff almost exclusively. There are some other projects we have that are funded by other groups and federal agencies, but we are extremely grateful, particularly to North Central and USB and Checkoffs in general, for all of the soybean cyst nematode funding. And it used to be the case that we had to introduce soybean cyst nematode every time, but I think most of you here are going to know all about the fact that it loses a lot of money for a lot of people and that we don't have good chemical controls. We're reliant on resistant soybeans, but they don't always work for everyone, and they seem to be getting a little less effective. And so I'm going to talk about the science of why we think that is happening and also about what we're trying to do about it. So I'm going to talk in more of a general term about our genome projects, and then Andrew's going to talk a little bit more about the genes themselves and how they're applied. Before I do that, though, we do have another project I wanted to mention. So traditionally, you'd go to your cooperative extension at your land-grant university, et cetera, to get more information about this and get latest info. That's still your best source. You've probably heard a lot about ChatGPT and things like that. And if you're like me, you're a little skeptical of the kind of things you get back from ChatGPT and sometimes also from Google. One of our projects with the AI Farms Center, which is a USDA NIFA funded AI institute on campus in the Center for Digital Ag, is we're trying to put together an advanced version of the ChatGPT chatbot that relies only on data that we know comes from state cooperative extension programs and that we trust. And you can check this out there. I may do a demo of it if I have time, but I'm going to move along in the interest of time. But if you go to that URL there, www.uiuc.chat, there's actually a link on that landing page to the crop wizard, and it will answer questions, for example, about soybean cyst nematode, and it will give you those references, 1, 2, 3, etc. Those will link you out to the original extension publications. So you don't have to trust what it tells you. You can go straight to the source and figure out where it came from. But unlike Google, it'll format this in a nice way and give you a narrative around it. So having said that, I'm going to talk about genomes. So human genome, one human egg cell contains 3 billion letters from mom, 3 billion letters from dad, and that is enough information to create typical humans, such as those you see on the right there. And all the information biologically needed to produce all the parts of the human body comes from that one set of instructions in that one cell. And the same is true of soybeans, and the same is true of soybean cyst nematodes. So human genome takes about 3 billion letters, soybean takes about 1.1 billion, wheat takes about 17 billion. So crops and people carry a similar amount of genetic information, and we're not always sure what most of it does. I'm going to tell you about our progress on the soybean cyst nematode genome and what we think that does. So one of the things that I mentioned briefly on the last slide is we talk about doing the human genome. The human genome is not the genome really of anybody. It's sort of a mixture of different people who donated samples to the project. What we're trying to do with soybean cyst nematode is create what we call a pangenome. And that's a picture not of one nematode or not of a mixture of nematodes, but a picture of all of the variation, all of the genes within soybean cyst nematode. It turns out that this is a bigger undertaking than we thought it would be when we started, but our original target was to sequence high quality genomes similar to the human genome and the soybean genome from all of the HD types that are defined by the soybean indicator lines, which I'll talk about in a little bit. And on that basis, Iowa State and us started a long time ago with DOE sequencing the TN10 isolate from Terry Niblack's group in Illinois. That started out as a type 0, and it has shifted a little bit through time. That was our original reference genome. That took a long time to do, and we really had to wait till the technology got better to do it. But more recently, we've sequenced these other isolates, which are from the other soybean cyst nematode types, but they're just one isolate of each type, right? So you can see here that the sum line, that's about 115 to 125 million bases. So it's quite a bit smaller than a soybean, but it's a smaller thing. And there seems to be a little bit less extra DNA in there than soybean, which I'll talk about in a bit, because there's still plenty of genes. However, if you look at those highlighted numbers over on the right, you can see our original effort there has a 60% that's highlighted, and then the others go up to like 64 or so. That's a measure that we use for genome quality, and it's a percentage of the genes that are thought to be needed for an organism to survive. So how come SCN has only got 60 to, well, we think actually about 64% now, because there was some missing in that one that has 60. That was our first effort. And also that size is probably a bit wrong. But still, it's only got 65% of the genes needed to survive. That's because it's completely dependent on soybean to survive. It can't survive on its own. So it's lost some genes, and it borrows some of those genes and some of those functions from the soybean. So this may be a way to address soybean cyst nematode and to get at it, but we don't know yet. So we're interested in what those genes are, what genes does it have, what genes is it missing, and what's happening to those genes as it evolves in these different HG types in response to people planting different soybeans. And we can compare these genomes, and it turns out there's a lot of differences. So I'm not going to explain this map here, but basically all those green dots are where they're similar. And so there's a lot of differences throughout these different soybean lines. And we don't think that differences in size are as huge as we thought initially. That published one is a bit big, but still we've got 10 million letters difference between these genomes. So there's a lot of difference between these different cyst nematode genomes. And each one of them contains something like 20,000 up to maybe 29,000 genes. So the actual number of genes in there is not that much different from soybeans. The genes are packed in tighter than they are in soybeans. And so then we started looking at the pan genome. So what genes are completely conserved in all of those and what genes are different between these lines? It turns out that quite a lot of genes are different between these lines. So we thought, let's start off looking at those 1,500 or so genes that look like they're exactly the same. And they're part of those 60 or so % that are thought to be necessary for life. But it turns out when we look at those, a lot of those genes are actually different. And a lot of those genes seem to show signatures of change between the different HG types. And when I say a lot, all of these different biological processes seem to have genes that are changing between the HG types in them, even among those genes that are close enough between the HG types that we can actually compare them. So in other words, this turns out to be a lot more complicated than we originally thought, but there are still some clues coming out of this analysis that allow us to narrow this down to a few processes that are probably particularly important. Melissa Mitchum at University of Missouri at the time, where Andrew is, and she's now at University of Georgia, made these populations of cyst nematodes by growing the same isolate of nematodes on different soybean varieties, different resistance sources. And one of the things that she in particular found, that others have also found, is that you can put almost any nematodes on almost any soybean, and if you give them long enough, they will come to be virulent on that soybean, even if they're not to begin with. So when we talk about these HG types, this is why we don't use the word races anymore, because races kind of implies that they're different groups that don't interchange. And really what there is there's a whole population of soybeans out of soybean cyst nematode out there. There's not all that big a population of soybeans, as I'll mention in a minute. So what happens is that huge population of nematodes, within any one nematode population, there's probably a few that can grow and reproduce on your supposedly resistant soybeans. So after a few generations, those are going to multiply and you're going to end up with a lot more nematodes than you started with. And so one of the things we did is we've developed a technology where, and these were adapted for 16 years, so it was quite a long time. We've developed a technology where we can sequence genomes from individual nematodes. So we can pluck out a worm from the population now and get a whole genome sequence from one worm. And this, just in a few years, we've gone from having to centrifuge a giant test tube full of egg pellets to get enough to do part of a genome to getting a whole genome from one worm. When we started doing whole genomes from individual worms, instead of extracting our DNA from millions of eggs at a time, we started to realize that all those worms are very different. And again, this is a very complicated story, but we can summarize it in a chart like this one at the right here. And the two populations that we have here, MM1 and MM2 from Melissa Mitchum's group, were adapted on different resistance sources from the same original population. And you can see that over time, those two populations have moved apart. So we've got a green cloud of points, which is the soybean cyst nematodes that can grow on one resistance source. And we got a blue cloud of points, which is the soybean cyst nematodes that can grow on the other resistance source. And there's really no overlap between them. But yet these were both derived from the same original population. And if we sequenced enough of them, we would certainly find probably some in the blue group that could still belong to the green group and could still grow on the resistance source in the green group. Also, remember, these have been grown for 16 years in pots in a greenhouse. So you think about how much diversity there is in these nematodes here, and then expand that to a field, even a single field, you're talking about a whole lot of nematodes, most of which are different from each other, and which are constantly creating new opportunities to parasitize resistant soybeans. You can see there on the left, this is the number of SNPs, as we call them, single nucleotide polymorphisms, so differences in the genome. And in each 100,000 bases, you can see in many parts of the genome, there are over 1,000 differences in every 100,000. So in other words, there's differences all the way through the genome, even between these supposedly inbred soybean populations growing in a pot. That's very different from what we see in soybean. Soybean is a little limited in its diversity. The nematode is very, very powerful in the amount of different genotypes that it's able to bring to bear in overcoming resistance. So in order to try and address this, we've also started a soybean pangenome project. And this project has been funded by the US Department of Energy, and they're doing most of the sequencing. It's quite a bit more expensive than the soybean cyst nematode project. But DOE are going to guarantee that they're going to provide high quality genomes for 400 lines. Obviously, Andrew and I are starting with the SCN indicator lines, right? So, but one of the things I wanted to point out is the original soybean genome allowed us to identify some of the early big resistance genes, such as RHG1B. But even then, when we identified RHG1B, it turns out to be a very complicated story. So this is a figure from one of my group's papers showing that even within one field of one inbred soybean variety, there is quite a bit of variation in that copy number repeat that is RHG1. It's a complicated piece of the genome. It's not just one gene, it's 3 genes repeated around 10 times in RHG1B. And if you don't continually select for that cyst nematode resistance, you tend to lose it out of the population, which is why some cyst nematode resistant soybeans were not very resistant at one time. You know, the companies now use this and are able to produce consistent PI 88788 RHG1B type resistant soybeans. But perhaps because those are now everywhere, we're starting to see a lot of virulence on that type of soybean. So that's the kind of thing we hope to be able to do for a lot of new genes using this pangenome project in soybean. So we have, as I mentioned, the 7 indicator lines that define those 7 HG types of SCN that we've already sequenced. And we're going to do high quality reference genomes of all of those lines. And hopefully that's going to allow us to narrow down both the location and the function of more and more resistance genes that we can incorporate into the population. And groups like Andrew's haven't been waiting for these genomes. They've been using positional genetics and other methods to narrow down these genes so that they can already breed with them. But hopefully this is going to give us some insight into how they work and how long they might last. So I wanted to point this out to you. This is a calculation we do from soybean, early soybean pangenome work. And that number there, NE, that's the effective size of the population of soybeans. Now, if you're in conservation ecology and you're looking at endangered species, anything less than a population size of 50, you worry that that organism is going to die out soon. And you can see that within the soybean breeding populations, particularly some of the older populations, we had numbers in the range of 8 to 13 for that effective population size. There's very, very little diversity in these north central region soybean breeding populations. More recent work has increased the size of that. So we've increased the diversity by purposefully bringing in exotic material, but it's still only going up, according to these kinds of measures, into the 30s and 40s. So there's not a lot of different soybeans out there to breed from. And to some extent, this is a limitation of soybean itself. It's not a highly diverse crop like corn is. If you compare that with soybean cyst nematode, and these numbers are subject to change, we're looking from those, those are all the soybeans that are bred in Midwest public breeding programs. This is all the nematodes that are present in that pot in Melissa Mitchum's greenhouse. And we're looking at that total population being somewhere in at least the 100,000s, probably into the millions. So even though there's a lot of soybeans in the field, they're very similar to one another. And so really the soybean cyst nematodes outnumber the soybeans, and it's not a fair fight. So we have to find ways to even up that fight via finding the most effective resistance sources and incorporating those resistance sources into nematode resistant varieties that are high yielding, which means we then have to reuse a lot of that low diversity material because that's the stuff that yields well in our region, and that's what's adapted. So I'm going to stop here because I know Andrew's got a lot to tell you about and take questions. I just wanted to point out that the people who actually did the work on the right, Rick Mason-Brink, Kim Walden, Essie Miraez, Lucas Borges, and Tongan Lee, those guys did this work, and then those PIs on the top left, we're the ones that take the credit for it. And there's several other people that were involved, especially in Melissa's lab. I'm afraid I don't have pictures of all those guys, and they're probably thankful for that, though. So I can take questions now, and then I'll turn it over to Andrew.
Doesn't look like we have any right now in the Q&A, but if anyone has any, they're welcome to put those in.
Would you like me to do a demo of the chat wizard if there's no questions?
Actually, yes, if you could do that. And then if anyone has any questions, they can put that in while you are doing that demo.
Okay. So this is based off of GPT-4 Vision, which is actually a very new version of ChatGPT that you actually have to pay for, but we're actually paying for that through the center. And if we ask it a question, let's say, for example, how can genome sequencing help with the problem of soybean cyst nematode? It's going to take a little while to think about that one, and it may not have a lot on genome sequencing because it is trained mostly on cooperative extension material. But oh, here it goes. No, by sequencing the genomes of both the plant and the nematode, we can gain insights into, yep, it's doing pretty well there. So it's quite good at these kinds of complicated questions that Google often gives you a completely off base answer to, because it can't go that far off base because it's trained on extension materials. You can see here that we've got, it'll give you these references here. So like we can go to number 10 there, and that'll show you exactly where it got that information from. It goes to the right page of this extension document from Terry Niblack for the Illinois Agronomy Handbook, but it doesn't only use Illinois information, it uses everything we can get from all of the land grant extension websites. So biocontrol strategies, for example, is going to MSU, and it's actually got that information from a grant description that people are working on, because we've asked it a kind of researchy question there. So it doesn't purely use extension publications, it scrapes land-grant university websites. And we think it collates that information fairly well. So it's part of this system that our engineering college has set up called UIUC.chat, where we're building more trustworthy AI. So trustworthy chatbots in particular is the idea here. And you can see it's UIUC.chat/crop-wizard. So this particular one is called crop wizard. If you go to the just UIUC.chat, you'll see that there's a few different things. There's an astronomy one. There's one on Clouder, which is a software platform. But here's crop wizard here. So we can just click on this and you can ask it your questions. So if anybody's got any questions for crop wizard, I can pop them in there, or I can just put the URL in the chat. Otherwise, I can hand you over to Andrew, and then you can ask crop wizard your questions instead of me. All right, I'm going to let Andrew get going because I know he's got a lot of stuff.
I was gonna say, it does look like we have one question in the chat. Does this chatbot try to make sure it grabs the most up-to-date extension information? For example, did it provide the updated information from the most recently updated agronomy handbook?
So this is one of its limitations. So it does get updated, but it doesn't automatically grab the latest of everything. It has to be manually updated at the moment. So in terms of being up to date, Google is probably better. So a little bit like ChatGPT itself, it's trained on a body of text up to a certain date. And then we have to issue sort of a 2.0 and a 3.0 with updated bodies of text. So I wouldn't guarantee it being up to date, but I would guarantee that it is more reliable and trustworthy than some other sources you could go to. So I'll just put that link in the main chat there.
And it doesn't look like there's any more questions right now, but if anyone has additional questions that they think of for Dr. Hudson, feel free to put them in there, and then we can hopefully address them at the end of Dr. Skabu's presentation. I'll go ahead and turn it over to you.
Good, thanks so much. That was such a nice presentation, Matt. Let's see. Sorry. This. Let me try this first. Okay, does that look good for everyone?
Yes.
Good. All right. Yeah, really nice introduction, I think, there from Matt. I don't the chat GPT, I love that concept, Matt. I wonder if they'll need us to talk about have a webinar anymore if we have. But I think the important thing maybe most people don't understand, I think that we're proud of, people like Matt and myself in academics and in scientific research, is that maybe you notice the training data that supplies these chatbots are produced by us, which is, I think, important to note. So thanks, Matt. That's really, really nice. So I'm going to continue on with Soybean Cyst Nematode, and I'm going to switch to talking about how we can fight Soybean Cyst Nematode on the plant side. So we have these genes in the nematode that Matt talked about, and those genes that the nematode has allow the nematode to infect SCN. And then we have the genes on the plant side, and those genes try to fight those nematodes and allow them not to infect the plant. So that's what those are the genes we're interested in breeding and developing new cultivars for farmers. And I want to start by, I'm going to talk about several different research areas And I want to highlight Dr. Mariola Lasowski. She's our molecular breeding research scientist here at the University of Missouri, and she does a lot of great research on soybean cyst nematode. Clinton Meinhardt and Bish is our lab manager for our entire breeding program, also an expert in soybean cyst nematode phenotyping. Bishnu currently leads our phenotyping lab. I'm going to talk about some work from Paul and Bassnett. He was a PhD student with us. And then we collaborate a lot with Dr. Melissa Mitchum at the University of Georgia. And I have to say, you know, all of the, as Matt mentioned, this research is supported by farmers. 99.9% of all the research funding that we receive is from checkoff dollars. I'm the PI of several grants with the Missouri Soybean Merchandising Council, as well as Matt touched on the NCSRP project. This project was originally started by Dr. Thomas Bond at Iowa State. Melissa Mitchum is part of that, Greg Tilka, Matt is part of that work, and Brian Deers, the soybean breeder at the University of Illinois. He recently retired, and we now have Eliana Montverde there. And then we have two projects with the United Soybean Board. One is led by Xinglu Li, and one is led by Andrew Bent at Wisconsin. So I'm not going to spend too much time on this. As Matt mentioned, I think most of us know soybean cyst nematode while we're here. The important thing I want to point out here is this is how we count nematodes. So we count the cysts under a microscope on a petri dish, and that's how we phenotype the amount of cysts that are formed on an individual soybean plant. And soybean cyst nematode, not only from a distribution point, this highlights the first introduction back in the 50s all the way up to 2014. And it's spread across the entire Midwest, and most all of modern soybean cultivation has to deal with soybean cyst nematode, but not, you know, this is just a distribution where soybean cyst nematodes are found. What we're dealing with now, as Matt mentioned, is a change in virulence. So SCN is present, but now it's becoming more virulent, meaning it's able to overcome the current types of plant genetic resistance that we use. And one of the reasons is the life cycle. The life cycle of soybean cyst nematode, from the eggs hatching all the way to cysts on the roots, is less than 30 days. So you can have multiple life cycles throughout a growing season. And as our winters are warmer, and as farmers are planting earlier and earlier, this can become much more of an issue with the number of actual life cycles that nematodes go through in a growing season. And so we manage nematodes currently a few different ways. We use seed treatments, we use resistant cultivars, and we use crop rotation. But each one of these has an issue. So seed treatments are really good in the beginning, right? The beginning of the season. So you don't get season-long protection from nematodes from the majority of seed treatments. And again, you have multiple life cycles, multiple generations of nematodes throughout the year. You're really only combating maybe that first generation with most seed treatments. And then crop rotation is very important. One of the most effective management strategies is to rotate crops, but that really only reduces your inoculum from season to season. It doesn't really help with shifts in virulence. And then we have resistant cultivars. The problem that we've had here is that we've overused the current RHG1B that Matt's group found and has worked on for quite a long time, along with Andrew Bent and Brian. That source has been overused, so it's starting to break down. And so Matt briefly mentioned on HG type. I want to go through it just one more time because it's really important for this presentation. So we determine HG type through testing the soybean cyst nematode population with 7 indicator lines, meaning 7 different soybean germplasm accessions. And we label those 1 through 7. So if that population has a has a percentage of nematodes that can overcome that type of resistance by more than 10%, then we label that with a number associated with the indicator line. So in this example, this soybean cyst nematode population that was sampled in the field, it had a female index of 15 on Peking, 35 on 88788, and then so on down the line. So we would label this SCN population HG type 1257, meaning it is reproduces on indicator line 1, 2, 5, and 7. This is different from what we would call an HG type 0, meaning there's not greater than 10% female index on any of the indicator lines. What's important for our farmers and everyone listening is that when we have an HG type 1257, that's mean more than 10% of those nematodes overcome both types of resistance that we currently have, Peking type resistance as well as 88788 type resistance. So in Missouri, these data that I'm presenting here are averaged over the last 5 years in our worm lab. And these are the female index scores for three HG types that we use consistently to do genetic mapping as well as phenotyping. And as you can see, I hope you can see, again, a HG7 is a similar race as HG0. It only reproduces on the indicator line 7. But if farmers in your field have a HG257, that means that you have at least 10% of those nematodes overcome 88788 type resistance. The population that we use, 88% overcome this type of resistance. And if you move to the most virulent race that I'm going to talk about, the most virulent HG type, that is HG1257, we have 20% of those nematodes, 1 out of 5, can reproduce and overcome Peking type resistance. So the most recent survey in Missouri shows that for the most part, we really don't have appropriate nematode resistance in the majority of soybean cultivars because we've used that 88788 type resistance. And so 5 out of 8 regions also have a higher female index than 30%. And this is where we make the cutoff with resistance and susceptible. So 5 out of 8 regions in Missouri have nematode populations that are predominant and overcome 88788 type resistance to where those cultivars would not be labeled as resistant cultivars. And that's across the entire state. Here's just one more example on 88788. 100% of all of the nematodes that were sampled were able to reproduce more than 10% on 88788. So these nematodes have changed. They really have, and that's just not arguable anymore. So we have 2 types of resistance that I'm going to talk about. And I think most people talk about, most farmers and seed dealers talk about. That's 88788 type resistance and Peking type resistance. So one thing I think, and I think Matt would hopefully agree, one thing I think we want to start doing in the industry is talking about genes rather than sources, because these genes are really what provides resistance to the nematodes. And if a gene's functionality is the same, or the same gene in multiple sources, then it doesn't matter what source it comes from, because that gene's functionality is what's determining resistance. So from here on, I'm going to talk about genes. The gene that's used, a single gene, a single locus that has a 3-gene tandem repeat that Matt mentioned is RHG1B. The Peking type resistance, this is actually mediated by 3 genes, and that's a point I want to get across in this presentation. So when you talk about Peking type resistance, that is mediated by 3 total genes, and that's at a minimum. I will also say that that's what we know as of today, and that we can talk about. So, and those genes are RHG1A, RHG2, and RHG4. So our first research question was, we have this RHG1B gene. What genes can we stack in 9763 to gain resistance? And we published this a few years ago now. Paul Bassnett was my PhD student. This is a picture of how we phenotype our material here at the University of Missouri. So each one of these plants is put into a little tube. This is a water bath that controls the temperature. We water every day. This lab has, our worm lab has been watered every day for the last 7 years. That's including Thanksgiving and Christmas and New Year's. Every day these nematode populations are maintained. Here is, we inoculate each individual tube, and then we measure the nematodes, the cysts that are formed on each of those. So what we found using 9763 is that for our most virulent nematode populations, this is HG1257. So these are the nematodes that can overcome Peking type resistance and 88788 type resistance. The two major genes that are involved in the plant providing resistance is RHG2 and RHG1. What I want to point out is that you don't see a peak here on chromosome 8 where RHG4 is. So RHG4 is actually a minor gene relative to conferring resistance to the major virulent HG types. It's really about this epistatic effect of RHG2 and RHG1A. And so the next slides here are going to be set up the same way. This is female index on the left, on their y-axis. And then on the right, I have the specific allele combinations and how those alleles and genes confer resistance in these different nematode populations. The first 4 are using RHG1A. The second 4 are using RHG1B. So if we have our HG7 population, this is, for this presentation, the least virulent nematode population. Again, this population is largely not found in the Midwest anymore, but this is where this RHG1B 88788 type resistance really works very well. And that's why it's been used for so long. The other combination that is important is RHG1A plus RHG4. That is another combination that really controls HG7 very well. And then what I also want to point out is that RHG2 and RHG1A, these do not control resistance. They are largely, if you have only those 2 genes, you do not get resistance to this least virulent population. But that's not what's in farmers' fields now. It is this HD257 and HD1257. And that's where RHG1A and RHG2 work to control nematodes, to drop below 30% female index. You just need these 2 genes. When you stack RHG1A and RHG2 with additional RHG4, this is where you really have really high resistance control of HG257. So this, again, is the 3-gene stack that resides in Peking type resistance. And that's why this works so well. So for our most virulent nematode populations, that is nematodes that can overcome Peking and can overcome 88788 type resistance, RHG1B combinations are largely ineffective. I know Matt's group and Andrew, there are other genes that we've worked with that can be stacked with RHG1B, but we're just talking about RHG2 and RHG4. And that really does have no effect on RHG1B type resistance. It's really about this epistatic effect between RHG1A and RHG2. And most importantly, as I mentioned, or importantly, in this particular highly virulent populations, the addition of RHG4 did not have as dramatic of an effect. So it's really about RHG1A and RHG2 driving this resistance. So in summary, that work really highlighted that Peking type resistance is 3 genes. If you're a farmer and you're having a problem with nematodes, you have virulent nematodes, high populations, and you ask for Peking type resistance, make sure it has all 3 of these genes, or these 2 at a minimum. So then our next question was, well, what about that 20%, right? What about this 20% of nematodes right here, right? So you may have seen 9763 and 437654 in our material, the populations that we use, that you don't see any cysts on those. Those are the most highly resistant indicator lines. But Peking, you do see about 20% increase. 1 out of 5 nematodes in this HG1257 will overcome Peking type resistance. So we thought, why don't we make some crosses and determine what is the gene in the plant that controls that 20%? And that's a recent publication led by Mariola within our group, and Vanavi and Melissa Mitchum's group. And I'm going to go through several slides here, but I want to start by saying we discovered a new gene, and it's called GM-SNAP02. So this GM-SNAP02 is the gene that controls that 20%. It's the gene that Peking does not have that these other 2 indicator lines have. And what we did was we did genetic mapping. And we have, this is the chromosome 2 chromosome. We did some genetic mapping using 9763 exclusively. In this slide, we fine mapped this QTL, meaning we went from a Google image of the entire town down to a Google pinpoint on top of a few houses. And we determined one of those houses was actually this GM-SNAP02 gene. And what we discovered, actually using, with some help from a lot of people, including Max Group, genomic information on these indicator lines, we found that there was 2 different alleles. One in 9763 was the result of a very large insertion in the gene. And in 437654, it was a relatively smaller deletion in the gene. And it goes back to Matt's point, and I think our point of understanding genes and knowing that here we have 2 actually different alleles, 2 different haplotypes, but both of those haplotypes confer the same type of resistance. And so then we talked with Melissa Mitchum, one of the best scientists I've ever worked with. And what she did was, the first thing she did was she looked at when would be the best time to measure how this gene is regulated. And that relies on when do these nematodes infect the plant. So here is Peking, 3 days post-infection versus 5 days post-infection. And then we have 9763 and 437654. So at 3 days post-infection, you can see a few of these nematodes are starting to establish feeding sites. And at 5 days post-infection, you can see that they have established a feeding site and they are now getting fat. They're enlarging and they're going to turn into a cyst. So these white arrows are pointing to the 20% of nematodes on Peking that can overcome those 3 genes, right? And we do not see any of those type of nematodes in the other 2 indicator lines. So that determined 3 days post-inoculation was the best time, the best point in space and time to measure gene expression. And that's what Vanavi did. And what you see here is the mock is not infected plants. And then we have infected with SCN. And the important takeaway, this is 3 different biological reps. The important takeaway here is that in Peking, this gene is upregulated when the nematodes are infecting, meaning the plant senses infection and upregulates this gene. And the actual nematode, as Matt mentioned, these genes within the nematode is exploiting this gene for infecting the plant. And then the PIs, as well as near isogenic lines that don't have this gene, a non-functional copy, it is not overexpressed. And actually, in one case, it was downregulated. So this led to the assumption that this was a non-functional mutation. So then Melissa Mitchum went in and used CRISPR technology. to knock this gene out in the plant. And so on the top here, you have two different biological reps. This is showing the green fluorescence shows that the plants were successfully CRISPR transformed, and they also have nice root development, so there was no issues with the roots. And then here is the empty vector control. So this is a functional SNAP O2, and you can see a mean average of a little over 80 nematodes per plant. And when we knocked this gene out, we had a highly significant reduction in the number of cysts. So that leads to the conclusion that this gene is a non-functional mutation. So if you stop this gene from working within the plant, you'll gain resistance. But the important thing is, how much resistance do you gain, right? What's the effect? So I want to say once again, this gene does not contribute to this HG7 type of resistance, right? So again, but this is the type of population that you do not find in farmers' fields anymore. What you do find is HG257, and this is where we increase the resistance, meaning this is the same setup as the same. We have female index, and then we have our allele combinations. So here's just our RHG1A and RHG2. When we include this non-functional SNAP mutation, then we reduce almost to zero. Generally, usually it's around zero for 257, and that was shown to be a significant reduction. And then we also have the same type of phenotype with our 1257. So we go from just this 2-gene stack to around 15 to 20% female index, and we drop that to zero when we add SNAP-O2. Okay, so this is Mariola and I and our team, we call this our breeding recipe. So if you're going to take a screenshot, well, actually this is recorded, I think. So this is really how we see, the first thing I want to these data are using the populations that we have here in Missouri. So if you have a red circle with a line through it, that is a susceptible phenotype, meaning it's greater than 60% female index. If you have an orange check mark, you're greater than 10%, but you're less than 30% female index. And we determine that as moderate resistance. And then if you're less than 10% female index, then that is our highly resistant varieties. So with just RHG1B, and then we have our HG types. So this is our least virulent. This is the population you don't find much anymore in farmers' fields. This is the population that overcomes 88788 type resistance. This is the population that overcomes both Peking and 88788 type resistance. So with only RHG1B, you only get resistance to this first HG type. Very similar type resistance with only RHG1A and RHG4. You only get resistance to this first one. When you have the 2-gene stack of RHG1A and this important gene we talked about, RHG2, that is when you get moderate resistant to both of the more virulent nematode populations. And then when you get the 3-gene stack, remember, this 3-gene stack right here is the 3 genes that are found in Peking type resistance. Then you get broad resistance to all 3 HG types. Now, when you stack gene SNAP-O2, the new gene that we discovered that I talked about, you gain resistance, high resistance to our virulent nematode populations. And then our 4-gene stack, I don't know if there's any breeders on the call, but it is a 4-gene stack that you have to work with. That's when we get high resistance to all 3 of our HG types. So then am I doing okay on time? I got 5 minutes, okay. So our strategic rotations of resistant genes. This was a project that we started with NCSRP, and this was also led by Poland. Another great graduate student, I believe she's at Bayer now, Monica Penowit with Greg Tilka, Brian Deers, Melissa Mitchum, and Clinton, and myself. So what we did was we decided to develop a field experiment to measure these changes in virulence and the changes in nematode populations if we changed the genetic mode of action each year. And so this is really precision agriculture with genetics in my mind, using genetic rotation to manage population density as well as virulence. And this was the picture in Iowa. This is here in Missouri. So we inoculated these fields, and we put up a little barrier. We call these microplots because they're very small. We planted 30 soybeans in each row, 60 total. This was in Iowa, 2 locations here in Columbia, as well as over in Illinois. We had 3 pyramids, and we didn't talk about these glycine soja genes. Matt's group and Andrew Bent and Brian discovered these glycine soja nematode genes that are very beneficial, and as well as this chromosome 10 is another gene. And so we have a lot of genes right now that we understand, and we want to understand how do these different combinations work in rotation with each other. So this is unpublished data, but both Paul and Monica said they're going to put this out this year. So what this shows here is we have our egg density on the y-axis, and then we have the C columns represent continuous planting, and then the R columns represent rotations of different genetic sources. And what we can, what I hope you can see here is that if we continuously plant our 9763 PI, that really reduced our population egg density significantly and dramatically compared to all other continuous planting. But that is because this has so many resistance genes in this one PI. But then the other thing to note here is that in our rotation scheme, when we've rotated between 8 different genes, meaning 4 genes one summer and then 4 different genes the next summer, then we really also reduced our egg density. And this is over 4 years. And the other dramatic thing in the data that we're seeing is that even though we controlled, I'll go back just for a second, you can see PI 9763 controlled egg density the most here in continuous planting. But if you look over here, we took those nematodes now and we inoculated 9763 and measured the virulence, and the virulence of those nematodes dramatically increased within just 4 years. And it goes back to Matt's point of the last slide was such a great slide of how quickly these nematodes can overcome most any type of virulence that we deploy. So, but then the other very dramatic thing that you might see is that if we rotated, this is where we helped keep that virulence from exploding. So yes, you can reduce it dramatically with 9763, but rotating the genetic mode of action each year will help with maintain virulence. That's what I said here in the summary. I'll keep it up just for a few minutes because a few seconds, because that was recorded. And I want to thank everybody. This is Vishnu here. Here's Mariola. Here's Clinton, Dr. Mitchum in the field, not in the lab, in the field. A bunch of people from our other group. Here's Vanavi. So thanks, and I'll be happy to take any questions from anyone.
Thank you, Andrew. And I don't see any questions right now in the Q&A. Oh, we just have one coming in. So if you have any questions, please go ahead and put them in there so we can make sure to cover the questions. And if we don't get to some of them, we'll try to address them later. But here is one that's on here. Is there any yield drag with this new gene combination?
So the first answer to that is I don't know. And that is always the first question, right? That is always the first question. So our current research over the next 2 to 3 years will be addressing that very question. I will, yeah, so I think I'll leave it there.
I'll just weigh in on that one a little bit. It's that Part of the problem for us as academics is that we have these lovely experimental farms with these tiny little plots. But in order to do real yield trials that would really reflect what you'd get broad acreage, that's quite expensive for us to do. So the companies do a lot of that, but that really takes a lot of land and a lot of people. So we don't like to put our money on the line and say, no, there's no yield drag, because there might be.
That's exactly right. And also to that question, I will add, there's some really nice research from Greg Tilke over the last 3 to 5 years, really showing that in a highly infested, large population density, virulent nematodes, and those type of fields, there's now a measurable benefit to having alternate Peking type resistance versus 88788. So, it's a double-edged sword.
Got
right,
another question. Sorry.
Yep, there's another question. The best rotation is currently with corn and then with Peking and then PI 88788, i.e. not doing Peking continuously. I guess if that's confirmation.
That is absolutely correct.
Okay. So I did have a question, I guess, for me, just how do you see this information that you all are working on these projects changing the way that farmers choose varieties and choose the resistant sources? I mean, obviously with this, we have is it hope that they will do that, or do we expect need to do that?
You want to take that, Matt?
I mean, I'll take a shot. I mean, I think unlike the situation with, say, corn rootworm, where by not planting a refuge, for example, and not rotating, you really create problems for everyone else as well as yourself. Because the nematodes don't move around much, if you're happy with what you're getting, it probably isn't going to damage soybean agriculture globally if you continue to grow PI 88788, because your nematodes have probably already overcome that resistance source, and continuing to plant it is only going to hurt your yields. So it's obviously, if that's still what financially works best for you and you've tried out different varieties, then, and there is still clearly a benefit in growing 88788 RHG1B type soybeans versus soybeans without any resistance, right? So you'll still see a gain from having those against, an older variety that doesn't have any resistance at all. But you'll getting to the point now where if you can find a good yielding variety with the full set of genes from the Peking type resistance that Andrew mentioned, you're probably going to see a significant gain from rotating that at least, every other year or so.
Yeah, and I would add, I think one way to look at it is over the last year or two, there has been a big push from the private companies to, in releasing more Peking type resistant in their portfolio. And I don't work for a company. I work for the University of Missouri, so I can't say for sure why that is, but you can think, I would say companies are investing in that and providing it for farmers because there is a problem, and because they have, because those varieties with Peking type resistance have been bred for much longer now, and they are showing a real benefit in certain fields and certain applications. So, I think that's an important point. When you see more Peking and more Phytophthora resistance in the newest varieties, I think that's one of the highlights of understanding that there is a problem.
All right, so we had a couple more things come in on the Q&A. Stephanie Porter, who's the Illinois Soybean Association Outreach Agronomist, she mentioned farmers are demanding more varieties with Peking. And then we have another question on here. I believe we'll probably do this as our last question. So if you do have additional questions, we do have Dr. Skaboo and Dr. Hudson's email addresses in the chat for you if you want to reach out to them afterwards with further questions. But we have, what is the reason that we don't see some of the other indicator lines used, like the ones not talked about today, i.e. 5 through 7 and Picket?
I guess I think that the main reason there is well, the reason they're not used is because it takes a lot of time, effort to breed with these PIs. That's the main reason, all of them. That's the main reason Peking resistance took longer to come to the market and have varieties, because you have more genes, more breeding effort, more linkage drag, all those things that are detrimental to agronomic traits when you're trying to intergrass a single gene. And I'll also say, as I mentioned from the beginning, I think it'd be really nice to move away from source. And it doesn't matter what sources it comes from, as long as that gene functions in the way that causes resistance.
All right, and we do have one more that came in on the chat. We'll go ahead and do that. With the population in the nematode world, so much population of the soybean world, and the replication ability of the nematode, I'm not entirely, can the battle be won? I believe is what
right.
that's asking.
Yeah, that's
I'll
a
let
good
Matt
question.
take that one.
So there's a famous analogy in plant pathology called the Red Queen from Alice in Wonderland, right? And if those of you familiar with the Alice in Wonderland story, the Red Queen, who's like a card with arms and legs, says you have to keep running just to stand still. So the battle probably cannot be won in the sense that we will not exterminate soybean cyst nematodes. They're not going to go away. They're here forever. If we keep running, which in this case means we keep developing new soybean varieties with new resistance sources, new genetics, and we keep improving our agronomy practices in terms of rotation and adding things like seed treatments where they help, then we might be able to stand still, right? So we may be able to keep the soybean cyst nematode at the level of problem it is now, maybe reduce it a little bit if we run really fast, but we have to keep running, and we're only going to just keep up with the nematodes if we do that.
What a nice way to end.
All right, we will go ahead and conclude today's webinar. Thank you again to both of our speakers, Dr. Andrew Skaboo and Dr. Matthew Hudson, and thank you all who were able to attend live. So stay tuned to lsoyadvisor.com for the recording of this webinar and for updates on ISA-funded soybean production research. And have a wonderful day. Thank you so much.
Thank you.
Thank you.